Thank you very much, Prof. Zamri. Uh, Now is the turn for Department of Physiology to have to talk on this on the topic on the herbal product and male sexual health. The talk will be given by Dr. Nelli Giribabu. First of all, let me introduce Dr. Giri. Dr. Giribabu is a senior lecturer at the Department of Physiology, Faculty of Medicine, University Malaya, and he's has done a lot of work in the field of reproductive and endocrinological sciences, and he has published more than 60 papers throughout his work here. And he's an expert in he's looking mainly on the herbal products and the female and male reproductive health. So without any further delay, I would like to invite Dr. Giri to deliver his talk. Please.
Good morning, respected delegates and professors and my fellow friends. Today, I would like to present one of my research topic on herbal products and male sexual health. So, before going to our real topic, I try to explain about male reproductive system. So, you know that male reproductive system consists of a complex system, and it have many tissues like a testis, epididymis, and other organs, and also different cells also there. The main male reproductive organ is the testis. The testis is composed by the seminiferous tubule. On the seminiferous tubule, we can see different type of the spermatogenic cells. Means the spermatogenesis occur on the serotolite cells, whereas other cells also we notice like serotolite cells. The serotolite cells can be supporting the testis, and also it can be protecting the mature sperm cells by attacking the immune cells and also attacking from the microenvironmental factors. Other cells also there, that is like Leydig cells. These are the main cells producing the testosterone. So you know that the testosterone produced by first the process is that cholesterol is transported into the inner mitochondria, then it is converted into pregnenolone, and it is converted into different hormones. Finally, the testosterone was producing. So the spermatogenesis and steroidogenesis controlled by the process of hypothalamus pituitary gonadal axis. I don't want to clearly go through the male reproductive system, but nowadays, so not only nowadays, that traditionally Malaysia using many herbal plants for the treating many diseases, like including male infertility. So traditionally, you know that the tonkatali, it is using for the sexual enhancement on the male, and also it can be improve the testosterone levels, and also it can be improve the erectile it can be reducing the erectile dysfunction. So besides the Kachi Fatima, I think hope most of the people using female reproductive complications like after menopause. Besides recent studies also the Kachi Fatima using for the treatment of male reproduction, like enhancing the testosterone levels. These medicinal plants nowadays Malaysia, a lot of Malaysia using to improving the sexual enhancement. So besides, we need a lot of medicinal plants are required. What is the reason the medicinal plants are required? So if you go to the clinic, if you look at the male infertility, very less synthetic drugs are available. So mostly if the vitamin C, vitamin D, some of the gonadotropic releasing hormone analogs, and some of the prolactin suppressors only using for the treatment of male infertility. But many drugs are need to find out to treat the male infertility. So in my study, some Malaysian medicinal plants and also some subcontinental, for example, South Asian countries like India, Malaysia, Thailand, these countries, some other plants also available. Those plants also can be useful for the male infertility. For example, the chlorophytum borevillineum, from the Ayurvedic time onwards, they're using these plants. So these plants have a they have the aphrodisiac effect and also sexual stimulant effect. So these plants will be traditionally using from India and also other subcontinental like the South Asian countries. Besides tornado diffuser, so this tornado diffuser also very useful for the improvement of libido, and also today I want to explain about other active bioactive compound like muriatic acid. So these medicinal plants and also these bioactive compounds have a different type of pharmacological activities like a anti-cancer, anti-diabetic, antimicrobial, and other pharmacological activities. Besides, these medicinal plants also have some of the potent effect on the male reproductive health. For example, C. borylinium or chlorophytum borylinium effect on the increase the sperm count and also improve the libido, and also improve the gonadot sperm motility, and also it can be improve the antioxidant engines and decrease the lipid peroxidation. So and also like a tornado diffuse also is useful for the treatment of male infertility by the improving population rate and improving the sexual motivation, and also it can be improve the nitric oxide stimulation pathway also. Also muriatic acid also in my study also it was proven that the sperm count and motility and viability also increase and steroidogenic markers also increasing and also oxidative stress also increased. So what is the gap? What is the problem statement? And now, these earlier study, the previous studies were discovered about these medicinal plant effect on the male infertility, but there is no prescribed mechanism was find out. So that if you find out the underlying mechanism, so it can be useful for the treatment for the male infertility. So nowadays the male infertility many drugs are needed. So because the very less availability of the synthetic drugs, especially like whenever you feel like some stress or male infertility caused by the male infertility, mostly the doctors prescribing vitamin C and some other drugs. So that we need to find out some alternative drugs to treat the male infertility. So here I want to try to explain my studies three different studies. The first of all, like in the study one, so I want to induce the oxidative stress by the hydrogen peroxide on the human sperm. So here the human sperm exposed by the 200 millimolar of hydrogen peroxide. So the reason the hydrogen peroxide can increase react to oxygen species very easily on the sperm, so that we can use hydrogen peroxide model. So after that, we're using a different concentration of the chlorophytum borylinium. And the second model, so the first we want to give the normal group, we're giving a normal saline. The second group, we give hydrogen peroxide 2%. Then the third group, the first 7 group, I given a hydrogen peroxide. After that, we treatment with the chlorophytum borylinium and also the positive drug like a dimethyl fumarate. So this is the NRF2 stimulator. Later I'll try to explain. So the third study was diabetic rats. So here I will try to use a type 2 diabetic rats. The first of all, the first 8 weeks, we try to give high fat diet so that the rats become obese. After that, on the 8th week, we give the low dose of streptozotocin. So whenever we giving low dose of streptozotocin, it will be mimicking a type 2 diabetes. Later, we will give 100 and 200 mg per kg body weight of tornado diffuser, and also we will give a bioactive compounds and 10 and 20 mg of muriatic acid. Sorry. So coming to my findings, so in the CB treated rats, so the sperm motility also increasing and sperm viability also increasing. From the picture we can see, so here the viable sperm means unstained. These are the unstained. The dead sperm means the pink stained sperm. So here we can understand the hydrogen peroxide treated rats, the sperm count and sperm motility also decreasing and viability also decreasing. HOS coil tile coil sperms also decreasing, and even though the sperm DNA fragmentation was increasing. Whereas treatment with the CB, this alteration was increasing, whereas the DNA fragmentation was decreasing. So from here we can see the picture like a HOS tile coil sperm was the tile coil was we can see here notice. So whereas the hydrogen peroxide treated rat like a 12 coil was not 12 coil properly, so that we can understand so the chlorophytum borivali can improve the hypoosmotic swelling, like whenever you exposing the sperm, so it will be become like a tail was coil behind. So we can see like a DNA fragmentation index was increased on the hydrogen peroxide rats. So here like a the orange color indicating like the sperm without hollow means here the fragmented sperm. The sperm with like the hollow, it is like a non-fragmented sperm. So here in the hydrogen peroxide treated rats, the DNA fragmentation was improved, whereas like chlorophytum borbolin treated rats, it was decreasing. So come back like oxidative stress. So you know that lipid peroxidation is one of the product of malonide. So it is indicator for the oxidative stress. So whenever the oxidative stress was increasing, before going to what is oxidative stress, so this is the imbalance between like reactoxin species and also antioxidant enzymes. So whenever the oxidative stress was increasing, so antioxidant enzymes like SOD, catalase, GPX enzymes was decreasing on the hydrogen peroxide treated rats, sorry, sperm, whereas it was improved on the chlorophytum borbolin treated human sperm. Besides, like NRF2, like a KEEP1 is a signaling pathway. I hope you know the NRF2 is a KEEP1 is like NRF2 is a translation factor. So it will be stimulating like antioxidant enzymes whenever the oxidative stress was higher. So NRF2 entered into the nucleus, so it will be attached to the ARE factor, and it will be stimulating like antioxidant enzymes and also NQ1 and HO1. So here in our studies, NQ1 decreasing and also KEEP1 also increasing. Other like antioxidant enzymes like NQ1, sorry, decreasing also. This alteration was improved by the chlorophytum borivalium. So from the immunofluorescence studies, we can understand. So here, the in the normal control rats, so here we can see the sperm is and the nucleus was presented by the NRF2, the protein. So whereas like the protein distribution was decreasing on the nucleus of the hydrogen peroxide treated human sperm. So besides like a chlorophytum borbolin treated like a human sperm. So these expression levels was increasing. So coming to the second study, so here on the second study, the mice was treated by the hydrogen peroxide first. Then after that, we will given orally chlorophytum borivalium different doses to the mice. So here we can see the sperm viability also like a decreasing, and also HOS12 also decreasing, whereas treatment with the CB it was improved. So here from the percentage of normal sperms, we can see normally like the mice sperm, they have a hook-like shape. But when we look at like hydrogen peroxide treated mice, so we can see different abnormal sperms. So it indicates that hydrogen peroxide can cause the oxidative stress. It will be lead to the different morphological abnormalities, and DNA fragmentation also improving. So so it was treated with whenever the treated with the CB, these alteration was decreasing. So coming to the histopathological structures of the testis, so here in the normal testis is composed by the different type of the sperm cells, and also the lumen also filled with the sperm. Whereas hydrogen peroxide treated mice, so we can see here the spermatogenesis was ceased, and also the lumen empty lumen was noticed. Whereas chlorophytum treated mice, these alterations was decreasing. So MDA, earlier I explained that MDA is one of the oxidative stress markers. So here in the hydrogen peroxide treated mice, we can see the brown color distribution was higher. So whereas chlorophytin borivalium and also DMF treated mice, these expressions levels was decreasing. So NRF2 and KEEP1 signaling pathway we can we perform with the immunofluorescence studies. Here we can see the NRF2 is a green color, the KEEP1 is a red color. So NRF2 higher we can see on the normal control, whereas in the hydrogen peroxide treated rats it was decreasing. whereas keep one is higher on the hydrogen peroxide treated rats. So whenever you treat it with the chlorophytum borvillianum, these alterations was happening. So besides, we can see the star protein. So the star protein is a steroidogenic marker protein. So it will just normally the star protein can be the transport the cholesterol from the outer side to the inner mitochondria. So the star protein expression was noticed on the leading cells. Here we in the leading cells of the hydrogen peroxide treated rats was we can see the very less expression than compared to the normal mice, normal rats, no, sorry, normal mice. But whereas chlorophytum borrowelin and positive control treated mice, these expression levels was increasing. So other studies like other proteins like connexin 43 and also the other steroidogenic markers like a 3 beta HSD. So we can see here the brown color like a connexin 43. This is like a the blood testis barrier markers, whereas 3 beta HSD is a steroidogenic markers. So these markers were also decreasing on the hydrogen peroxide treated mice, whereas it was improved in the CB treated mice. Second study, so earlier I explained about the second study, Ternode diffuser treated to the type 2 diabetic rats. So here, whenever you giving like a normal control, like a normal diet, so the up to the 12 weeks, it was maintained like a normal fasting blood glucose levels. Whereas on the 8 weeks, we try to give the low dose of streptozotocin. So we want to mimic the type 2 diabetes. So here the fasting blood glucose levels was improved. So whereas treatment with the CD, the TD, Ternode diffuser, so that fasting blood glucose levels was significantly decreased. And besides, so insulin levels was also decreasing the diabetic rats. So whereas insulin levels was improved on the TD treated rats. So from the right hand side, we can see like a different morphological abnormal sperms improved on the diabetic rats, and also sperm viability also decreasing, HOS also decreasing, and DNA fragmentation also increasing. Besides, the sperm count and motility also decreasing the diabetic rats, whereas the treatment with the TD, these alterations was reduced. So another coming to the this figure, so the TD effect on the oxidative stress and inflammatory markers. The left side is the oxidative stress markers, and right side is the inflammatory markers. From here, we can see like a NOx1 also improved on the TD diabetic rats, and also lipid peroxide also improved on the diabetic rats. Besides, so we can see the antioxidant enzymes like SOD, catalyzed GPX levels was decreasing in the diabetic rats, whereas TD treated rats, these levels was improved. So when you look at the right hand side, NFKB and IKKB, it is like the inflammatory markers. General condition NFKB, IKKB always present on the cytosol. So whenever the inflammation was improved or whenever oxidative stress was improved, NFKB translocate into the nucleus. It will be trigger other pro-inflammatory cytokines like interleukin 1B, interleukin 6, and TNF alpha. In our studies also we can see like inflammatory markers like NFKB and IKKB and also TNF alpha also improved in the diabetic rats, whereas treated with the TD, these levels was expression levels was decreased. So the third and final my findings coming to the myrcetic acid. Even though the myrcetic acid also we treated to the type 2 diabetic rats, and the type 2 diabetic rats, the sperm count, motility, viability also decreasing, and morphological abnormal sperms also increasing, whereas treatment with the myrcetic acid, these alterations was decreasing. Besides, we can see on the right hand side, the mRNA levels like NFKB and TNF alpha, interleukin 1B and INOS. So these levels was improved in the diabetic rats, whereas treatment with the myrcetic acid, these mRNA levels was decreasing. So from even though it was confirmed by the Western blot studies, even though it was confirmed by the immunisto and immunofluorescence studies also. Besides, we can see like the apoptosis marker, like BAX is a pro-apoptosis and BCL2 is anti-apoptosis, and caspase-9 levels was also the alteration was happen on the diabetic rats, whereas the myrcetic acid treatment with treated diabetic rats, the BAX levels was decreasing, whereas the BCL levels was improved. So, and caspase levels also decreasing. So it, and also it was proved by the immunohisto studies also. So whenever the oxidative stress was reducing on the diabetic rats, whenever like apoptosis also reducing in the diabetic rats, so that the steroid genesis was improved, so that the testosterone levels was also improved whenever in the mustard acid treated diabetic rats. So here from this figure we can understand these steroidogenic markers like a 3 beta HSD and like a coming to that D. So this is star proteins we can see on the leading cells, and also ARA54 also we can see on the leading cells, the red and green color. So in the diabetic rats, these levels was decreasing, and also IHC image also with the showing like a CYP11 450 and also decreasing the diabetic rats, and also CYP19A1 also decreasing the diabetic rats. These the steroidogenic markers levels was improved whenever the treatment with the mustard acid for the type 2 diabetic rats. Finally, from my studies, it can be discussed that so the by using the natural like a medicinal plants, like it is very good for the health, because whenever using like a synthetic drugs, so it will be sometimes it will be causing side effects. So by the using like a like a medicinal plants, so it is very safe for the human health. So especially like this, my study chlorophytum borvilianum, tornado diffuser, and mestic acid. These are the good like the natural products for the promising the protecting the sperm and also testis also to protecting against the oxidative stress and also inflammation and apoptosis induced by the hyperglycemia and also the hydrogen peroxide induced oxidative stress also. The overall my study it will be conclude that these medicinal plants and mustard acid also, these are the alternative therapies for the improving the male fertility, especially like a diabetes and with the oxidative stress. Finally, it will be concluded that these medicinal plants and bioactive compound, so these alternative therapy for the like a infertility, it can be improve the fertility by the protecting against the oxidative stress and also protecting against the hyperglycemia. So these are my latest one of the latest publications. So thank you, friends. So
Thank you very much, Dr. Giri, for the nice presentation. Now I would like to open the session to the participants. Any question from the participants? Uh, do you have any questions about the presentation by Dr. Giri on these natural products? So few of these are the selected natural products which he worked in his lab in trying to improve male fertility in disease conditions like diabetes and in oxidative stress. So apart from that, I think if you have no sessions, I would like to end the talk this morning. So thank you very much for attending and participating in the talk. Thank you.
thank you, friends. So I think one of the doctor they asking for the can there be any overdose while using these herbs? Doctor, can you hear my voice?
Questions there? Since the
Yeah,
question, okay.
yeah. So generally, when we going for the herbal drugs or medicinal plants, first we want to do like some of the preliminary studies, like acute and chronic toxic studies. So the acute and chronic toxic studies, so it will be proved that these medicinal plants are the safe. So there is no mortality. So that so not only these medicinal plants, any medicinal plant also, first we want to try the new medicinal plants with like the toxicity studies. So whenever the toxicity studies the prove these drugs are maybe they safe, then next step we're going for the different doses like a so different doses. From the different doses we want to select like a which dose maybe can improve like a, something like a the good result. So those dose will try to giving to the animals so that there is no like a the overdose. We cannot say like a overdose. So like we before going to the real like the treatment, we want to test like a trial run like a acute toxic studies, and also like a cell culture. Also we want to do like other studies also, so that these are the so we want to select the doses.
Okay. So any other questions to Dr. Giri? So if there's no question, I think that's a very good talk on this topic. So I would like to just conclude the talk today for today's session, and like to thank both speakers, Dr. Hassan and Dr. Nelli Giribabu, for the talk this morning.
Thank you, Prof.
Thank you very much.
Thank you.